Good morning, my name is Ingrid Campo and I am going to talk about dengue disease. First, we need to know about the definition of the disease. Dengue is an infectious viral disease transmitted by the bite of infected female mosquitoes, primarily Aedes aegypti. Signs and symptoms of dengue. It is characterized by high fever, several headaches, joint and muscle pain, and rash. It is not transmitted from person to person and it is common in tropical and subtropical areas. Warning signs. Seek immediately medical attention. These signs generally appear when the fever subsides between the third and the seventh day. Severe and continuous abdominal pain, persistent vomiting or vomiting blood, bleeding from the gums or nose or blood in the urine, dizziness, fading or change in level of alertness, lethargy, irritability, difficulty breathing. The family Favipidae is a large group of viral pathogens responsible for causing severe disease and mortality in humans and animals. The family consists of three genera: flavivirus, pestivirus, and epacvirus. The flavivirus, Latin flavus, meaning yellow because of the jaundice induced by yellow fever virus, comprise a large genus of medically important arthropod-transmitted enveloped virus with more than 70 members that include dengue virus. Symptoms of the flavivirus infection can range from mild fever and malaise to fatal encephalitis and hemorrhagic fever. Causative agent. Dengue virus is a single-strand positive sense RNA genome consisting of 11 kb in length. The single predictive polyprotein is translated by the genome that it is used by the virus encode protein into 7 non-structural proteins and 3 structural proteins, which are defined as follows: non-structural gene 1, NS1, non-structural gene 2A, NS2A, and non-structural gene 2B, NS2B, NS3, NS4A, NS4B, non-structural gene 5, NS5, and the structural proteins including capsid protein, envelope protein, and membrane protein. Some of the non-coding regions are also present on the 3' end of the genome. Virus attribute. Structure and genome. It is a positive sense single-strand RNA virus with a spherical envelope. Serotypes. There are 4 distinct jet-related serotypes: DENV1, DENV2, DENV3, and DENV4. Immunity. Infection with one serotype confers permanent immunity against that specific serotype, but only temporary and partial protection against the others. Reinfection and severity. A second infection with a different serotype increases the risk of the developing severe dengue. Pathogenicity. The virus primarily infects macrophages and monocytes replicating within white blood cells. The structural proteins play a dynamic role in the structure of viral proteins, as the non-structural protein plays a vital role in virus entry, replication, assembly, and pathogenesis in the host, where they cause disease and pathogenesis. Mechanisms of infection. Regarding DENV infection and replication, there are numerous mammalian cell receptors reported to be mediate DENV attachment and entry, such as DC sign, L sign, heparan sulfate, mannose receptor, laminin receptor, dopamine receptor, and others. In the early phase of DMV replication, virus particles would attach to the receptors and further enter the cell via the clarithin-mediated endocytosis pathway. The virus merges with the endosomal membrane once it has reached the interior of the cell, as is then released into the cytoplasm. When the virus particle splits apart, the viral genome is released. The viral genome is replicated after the translation of the viral RNA, vRNA, into a single polypeptide, which is then divided into 10 proteins. When structural proteins and freshly synthesized RNA protrude from the endoplasmic reticulum, virus assembly takes place on its surface. The trans-Golgi network is the place where the immature viral particles mature and change into their infection form. At that point, the full developed virus are expelled from the cell and are free to infect additional cells. Pathogenesis and virulence factors. Transmission and dissemination. The virus enters through a mosquito bite and initially replicates within dendritic cells and macrophages, subsequently disseminating through the lymphatic and circulatory systems. Antibody-dependent enhancement. In secondary infection involving a different serotype, pre-existing antibodies fail to neutralize the virus. Instead, they form complex that facilitates its entry into monocytes, thereby exponentially increasing viremia and the inflammatory response. Cytokine storm. Massive infection triggers an excessive release of inflammatory cytokines, tumor necrosis factor, IL-6, and others, leading to increased vascular permeability, plasma extravasation, and shock. Endothelial dysfunction. The virus infects or disrupts the endothelium, resulting in thrombocytopenia, low platelet count, and hemorrhage due to cellular apoptosis and vascular barrier dysfunction. Dengue virulence factors. NS1 protein, non-structural one, a K factor released into the bloodstream, it causes dysfunction of the endothelial glycocalyx, increases the vascular permeability and plasma leakage. Glycoprotein E, envelope, mediates the binding of the virus to host cells and membrane fusion, which is essential for infectivity. Protein M induces apoptosis in immune cells, thereby facilitating viral propagation. Evasion of the immune response. The virus inhibits host interferon production, delaying antiviral defense. Genomic variation. Certain strains are associated with higher virulence and hemorrhagic manifestations. Epidemiology. Dengue virus causes the disease and burdens in most tropical and subtropical regions of the world, mainly in the Caribbean, Central and Southwest Asia, and South America. More than 100 countries are affected by dengue virus all over the world each year, and there is a high risk of infection with approximately 3.6 million people live in these countries. Over the past 20 years, the number of dengue cases reported was 505, 430 cases in 2000 to over 2.4 million in 2010 and 5.2 million in 2019. The number of reported deaths increased from 960 in 2000 to 4,032 in 2015, mostly affecting younger age groups. In 2020 and 2021, both the overall number of the cases and the number of reported deaths appeared to be declining. The data is still incomplete, as the COVID-19 pandemic may have made it more difficult to report cases in some nations. Global incidence and prevalence. Annual case, it is estimated that there are between 50 and 400 million dengue infections per year. Population at risk. Approximately 4 billion people, nearly half of the world's population, live in endemic areas, primarily in tropical and subtropical regions and Asia, the Americas, Africa, and the Pacific. Trend. Incidence has increased significantly over the past 3 decades, accompanied by an increased disease burden that approximately doubled between 1919 and 2021. Groups and high risk of severe dengue. Secondary infection, reinfection. Individuals who are infected a second time with a serotype different from that of their initial infection face a significantly higher risk of developing severe symptoms. Children and infants, children under 15 years of age, particularly infants, are more prone to developing severe dengue due to the increased permeability of their microcirculation. Older adults, individuals over the age of 65 are high risk of complications. Transmission mechanism and vector. The principal vector is the Aedes aegypti mosquito in the primary vector of transmission in tropical and subtropical regions. Secondary vector is Aedes albopictus, can also transmit the virus. Transmission cycle. The mosquito becomes infected by biting an viremic person, one with the virus in their bloodstream, and following an incubation period of a few days within the insect transmit the virus to healthy individuals through its saliva. Vector abyss. Aedes aegypti lives inside homes in their immediate surroundings, preferring accumulate clean water in which to lay its eggs. Incubation period. Generally 4 to 10 days following the mosquito bites. Culture and diagnosis. The diagnosis of dengue is based on virological tests, RT-PCR, NS1 antigen during the first 5-7 days of the fever, and serological tests, IgM, IgG after the 7th day. Viral culture using mosquito, C636, or mammalian virus cells, confirm the infections by isolating the virus until it is time, consuming and used primarily in research settings. In summary, if the infection is very recent, 1-3 days, the NS1 test is the most appropriate, whereas after the first week, IgM is sought. Culture methods. Cell culture techniques are employed often utilizing high-density bioreactors to maximize production. These methods are rarely used for clinical diagnosis, Antigen tests or PCR are more commonly used instead. Viral replication. The virus lacks independent life and requires infecting a host cell in order to replicate, utilizing a key protein known as NS3 for this process. The cultivation of dengue virus is a complex process used in research to study viral replication, evaluate antivirals, or produce vaccines, typically employing mammalian cells such as virus cells or mosquito cell lines. Prevention and treatment. The mosquitoes that transmit dengue are activated during the day. To reduce the risk of the contracting dengue, protect yourself from mosquito bites by using clothing that covers as much of the body as possible. Mosquito nests, ideally treated with insect repellent. If sleeping during the day, window screens can be used. Treatment. Management dengue at home. First, hydration. Drink plenty of water, oral rehydration solution, or natural juice to replenish fluids. Medication. Take paracetamol for fever and pain following the doses recommended by a doctor. Rest. Observe absolute rest to allow the body to recover. Mosquito control. Use repellent and mosquito nets during the net febrile phase to avoid transmit the virus to others. Avoid aspirin and ibuprofen as they increase the risk of bleeding. Treatment of severe dengue. Severe dengue requires immediate hospitalization. Hospital treatment includes intravenous fluid and electrolyte replacement, continuous monitoring of blood pressure and vital signs. In severe cases, a blood transfusion may be necessary. Early and appropriate treatment drastically reduces the risk of mortality. There are some references that I use for this work. Thank you so much for your time.